Thank you. We're very pleased, delighted actually, to be here today with you, and we're really excited to show you the progress that Ventripoint has been making. We're into the third generation of this device, and we feel that this is much closer to the mark. As I was saying at the beginning, we listen to our customers, we listen to the people that we're working with. On various projects. We heard you, and as I said, we put our heads down and we got to work in revising the device. So I'm excited to show you the latest and greatest. And hopefully at the end of this presentation, we'll show you some things that we are working on to come in the future. So the problem that we are trying to solve with the Ventripoint technology is this concept around inconsistent, unreliable measurements with 2D and or a 3D echo, and really around this issue of image quality and disease state dependence, being able to take some of these images where oftentimes you're not able to rely on those measurements for a variety of reasons. It could be poor imaging or echogenicity of the patient themselves, or to do with the disease state itself and the progression and the technology that you're using, namely if you're using 3D echo. And so this is where Ventripoint comes in. So our solution is we are a diagnostic aid that was developed to provide a point of care solution to better communicate the heart structure and function without the need for MRI. The idea here is not to replace MRI. We are a tool that is to be used with 2D or 3D echo in cases where you may not want to use the MRI. There may be an issue with accessibility, or you can't use the MRI for a variety of reasons with your particular patient, specifically in the case of pediatrics, which is why the technology came to be in the first place. It combines the ingenuity of the cardiac MRI and the echo with the power of Ventripoint's proprietary AI technology, knowledge-based reconstruction, aka KBR, and we'll go into a little bit more detail and describe the KBR technology for you. It allows for 3D heart analysis from 2D ultrasound scans, as well as we do handle 3D echo and MRI part of the software. You're able to create a 3D model of all 4 chambers of the heart. And even with the complex shape of the right ventricle, VMS Plus gives you results that are equivalent to MRI based on how the technology, or rather the KBR, was developed. So what is the clinical value to you? Quantification and visualization. So what you are going to see today and a little bit later is some of the new approaches that we're using for quantifying and visualizing intricacies of heart motion for all heart diseases and disorders. That is the beauty of the VMS Plus. Wherever volumes and ejection fractions are warranted or desired, the system can be used. It has been validated for that and has regulatory market approvals in Canada, United States, and Europe for those purposes. It deals with the complexities of diseases to deliver new levels of clinical information when dealing with those complexities. And I think most importantly for the audience today is that clinical confidence with accurate assessment of cardiac anatomy, specifically around RV function, which we know that some of the tools today are lacking. So the key technology. Ventripoint leverages the benefits of 2D echo to allow for improved image quality, in the sense that even for those images that are not necessarily the perfect images, you're still able to get volumes and ejection fractions with the accuracy and reproducibility that you need in your practice for your patients, the care of your patients. It allows for this better temporal and spatial resolution, or rather resolving that problem that we know that you have with the technology in 3D echo. And the right ventricle is often difficult to be able to capture in that one 3D data set. That's okay. that's something that VMS Plus can handle, and we're hoping when we demonstrate the power of the technology to you in an actual demonstration, you'll be able to see that for yourself. You don't need to take our word for it. The underlying know-how is this algorithm that was developed by Ventripoint. It's a knowledge-based reconstruction a database. It reconstructs the patient's all four chambers of the heart. Once again, it does handle poor quality images, so it accepts sparse, unevenly distributed input data points. With the current technology that is out there, typically a contour is created around the region of interest, whereas with the Ventripoint technology, you don't have to see everything in the image. You collect your views as you normally would in any typical echo study, and you drop points for specific anatomic landmarks where you see those landmarks. If you don't see them on a view, you move on to the next view where you're able to see that landmark. Also, as I mentioned earlier, we do handle a 3D echo. It works the same way, but we take it back to the fundamentals. Rather than trying to get people to create this jigsaw puzzle of the 3D model, we allow you to slice this 3D model, create the 2D from which it was derived, and recreate that 3D model for you so that you can visually actually see that. Today, we're just going to be showing you the technology with the 2D, and we're happy within the future, anyone is interested, to show them a 3D echo and MRI package. So the technology is based off of this MRI image catalog. It is proprietary to Ventripoint. Many years to develop. It contains thousands of images that cover a broad spectrum of ventricle and atrium shapes and volumes as manifested in a variety of different clinical conditions. I'm actually going to turn it over to Desmond, our head of technology, to describe in a little bit more detail about how the KBR works at a high level, if you may, Des. Des? Okay, looks like he's not online. So essentially how the technology works is as you run your echo, there's position and orientation data that is transmitted from a set of sensors that are located on, so there's a patient sensor that's placed. As you can see, the arm is gone. We've completely removed the arm and we've replaced it with this little patient sensor that sits opposite the side of the heart, and another sensor that's placed onto the probe, onto the ultrasound probe. And while you're carrying out your echo study, the position orientation data is being transmitted to a little console that sits off to the side of the echo machine, and we'll be demonstrating that for you shortly. And that position and orientation data is input into the algorithm with the point placement. That data, what the KBR does is it searches through its MRI shape catalog and identifies different segments of different hearts to put them together to create this 3D model. The system is designed to coexist with your existing 2D system without complicated workflow. The idea was for us to integrate into your workflow, not for you to integrate into our workflow. So as I keep saying, there's an equivalency to MRI. We have validated studies, clinical trial studies that were conducted early on with the technology. This technology is about 10 years in the making to get to this point, and there is an excellent correlation with MRI to allow you to get those accurate results. And there is regulatory market approvals in Europe, United States, and Canada for that particular application. So the way that the system is, the workflow is split out is into image acquisition and image analysis. As part of the image acquisition, as I said, you use your standard 2D echo views to generate 3D models. So the entire system is set up to provide you with the navigation through. It doesn't require you to remember what you were trained to do. It actually provides visual as well as audible cues throughout the process. So one of the navigation screens that you will see is the views that need to be collected to be able to generate these 3D models and cardiac metrics. Keeping in mind that these are the views that are standard views that you would collect anyhow. There's nothing special here. What it is demonstrating on the screen to you is the different types of anatomic landmarks. As you can see, these little color-coded X's. They're for different landmarks that we hope you'll be able to capture in those particular views. So, for example, if you were collecting a parasternal long axis for the RV, this is what you would like to be able to see on the screen and capture that, and that would be input into the algorithm to generate that 3D model. So as specific views are being captured during the exam, the VMS Plus console will capture those transducer coordinates relative to the patient sensor, or rather the magnetic sensor that's located there. As you can see, you are able to collect for all 4 chambers of the heart. You can do each one individually, or you can group them. For example, if you want to do RV, RA, or LV, LA, you're not restricted. And the beauty of it is you go, you collect your echo study, and you click done at the end, and you are done. There's absolutely nothing else that is required of the user in the image acquisition. In terms of the image analysis, as I mentioned, it uses specific anatomical landmarks. So you see these X's are color-coded for the different anatomical landmarks that are required for the algorithm to be able to generate that 3D model. Keeping in mind, it doesn't matter what view you drop these points on. It only understands the points and how they are relative to one another when it creates that 3D model. So coming back to, if you don't see the point, you don't drop it. And it is a semi-automated approach that we've created. We are working towards a fully automated approach, but the way that this software was designed is we don't want to take the full control out of the users. So you don't have to rely on our templates to drop the points in the right spot. If you're not satisfied with it and you want to make adjustments, the software does allow for that. And these anatomic landmarks, they are specific ones that are assigned to each view. We are not asking for you to drop any points, that's anything that's unfamiliar that you wouldn't normally be looking at anyhow with these. We want to stress that we have really tried to integrate into your workflow and not add anything extra to what you are doing. So the key features of the VMS Plus is around the accuracy and the reproducibility. We do appreciate that you need to be able to trust the numbers that you receive from the software. And what we have been approved for is MRI equivalent volumes and ejection fractions every time. You use it on the same patient. Should you use it on the same patient, should you use it on different patients? The compatibility with 2D and 3D ecosystems on the market. In the past, you probably recall with the arm, there needs to be a calibration for every system that you plan to use it with. That is no longer the case. It is literally plug and play. You can move it from one ultrasound system to the next with ease, and there should be no issues. We have now a universal calibration. Standard echo views, once again, nothing special that's required of the sonographer in capturing those images. It is what you would capture during a standard exam. So what are the benefits of VMS Plus? Well, especially now, time is of the essence. So it's a reduction in the treatment time to achieve the same effect as cardiac MRI in cases where you want to get those quick results. Also in cases where, for example, in pediatrics, where you don't want to place the patient in an MRI. Also with patients where you don't want to place them in MRI for an extended period of time. It does handle sparse data. As I said, you don't need to create this perfect contour around the region of interest to be able to create that 3D model and derive those cardiac metrics. It's about dropping points where you see those anatomical landmarks on the views on which you see them. And it's the accuracy, once again, being able to trust those numbers. This is what we've heard time and time again from our customers. We have to be able to rely on those numbers. And so we've taken great efforts to ensure that. As you can see from multiple publications, there's over 60 publications that speak to that accuracy and reproducibility for the VMS Plus. There's a number of different other applications that we are expanding into, where there are studies that are they're in various phases of either we're starting them or they have started and they're just in progress. Things around blood pressure control, heart remodeling in pregnancy. That is a study that's actually underway in Canada, where they're going to be looking at pregnant patients during their pregnancy that had underlying cardiovascular conditions prior to pregnancy. They will be following them through pregnancy as well as post-pregnancy for, I believe, a year thereafter. And of course, as you know, in cancer therapy, there can be damage to the heart, so there needs to be a quick and easy way to be able to monitor these patients as they're undergoing chemotherapy. And this is where we think that the VMS Plus can be helpful. As I mentioned, there are multiple articles that have been published over the years speaking to the power of the VMS Plus, or rather the first generation of the device, which was known as the VMS. It gives you accurate results even with complex and unpredictable shape variations in congenital heart disorder patients with RV to PA conduits is one study that was carried out a number of years ago. It has excellent reproducibility even with complete retest by different examiners in patients with pulmonary hypertension, and it is superior conventional TV echo analysis in that regard. These are the conclusions that were derived from these papers. VMS analysis, 1/3 of the time it takes to analyze cardiac MRI study for RV function. It's a short learning curve now to establish this VMS technique. I know in the past with the arm, it was quite a challenge. You couldn't move the patient. Bells would be going off. That has all been removed. Now with the sensor, because it is localized to the patient itself, that is your datum point. You're able to move the patient as you normally would during your echo study. As I said, we really tried to make sure that we integrated into your workflow and didn't add any extra effort on your part. It is equivalent to cardiac MRI in accuracy and reproducibility in patients. It's been demonstrated with pulmonary hypertension with excellent inter-observer variability, and it gives accurate volumes for RV using 40 studies and takes 5 minutes to perform in normals and CHD patients. So there's nothing extra that's added on the acquisition side. The only thing is you have to place the patient sensor down, place the transducer sensor on the probe, and
let me find out how to move this thing out. Okay, so let's call up. Actually, this is the patient that I had on my IE33. This is the patient in our analysis application. So what we see here are all the clips that were acquired for doing, in this particular instance, for this particular instance, it was for an RV, but I also acquired images for the LV concurrent at the same time. So let's just go back to the RV. Right now, I can play them all. So our console interface showed you that we wanted a parasternal. We were interested in the RV inflow. I took a second parasternal because I was so proud of the first one. And then we did an RV outflow, short axis at the level of the aortic valve. Another one, short axis further down towards the distal. And a couple of 4 chambers. So let's pause. And so the first thing we do is we call up our cine. I have the ability to scroll by dragging my mouse, or I can use the mouse wheel. And what I'm looking for is end diastole or end systole. It does not matter where you want to start. So we'll start for end systole. I drag my template across. I have the ability to adjust that template, make it larger so it hones in. I can rotate it too. I can double click it and place it, and I'll just refine my tags a bit here. So we've done end systole for the peristernal. I scroll back to end diastole. Drag my template across. Align it a bit better. Double click and place it.
Also, Jim, if you wouldn't mind toggling the guide panel, just to show everyone that even if you don't want to use these templates, and you want to just place an individual anatomic landmark, you are able to do that as well.
Yeah. So what Alvira is alluding to is here. So we've automated this for you, but you also have the ability to, or the flexibility to select the anatomic structure of choice, and you can place them also this way, manually place them, you might say.
And this is coming back to if you've got a particular view where you can't see all of those, all of the points that you need to drop, you just drop the ones that you can see and you move on to the next view.
And then we can move forward. RV inflow view. End diastole. Pull my template across. Make it a little bit bigger. Rotate it a bit. Double click. I have a very good view of this anterior RV free wall, so I'm going to put 2. And just like I see it here, I'm going to put 2 on the inferior RV free wall. Pull my tricuspid tags. And I've done end diastole for the RV inflow. I scroll forward for end systole. Here's my smallest chamber size. Double click. Place my end systolic tags. Find my annulus. And we go forward through. So there's end diastole and systole. And then we do that for the RV outflow. We do it for the short axis at the level of the aortic valve. We were interested in the pulmonary, tricuspid, conal septal region, that anterior infundibular wall. At the short axis. And then finally, an apical forechamber.
And also, so Jim, if I could so really what we're doing, this is what we call sparse data. So you don't have to see the complete wall. If you can make the wall out in some cases, that could be enough for us. And this is the whole idea of it, some cases where the image is compromised for whatever reason, we still want to be able to get an accurate measurement. That's our sparse data statement. That's all. Over to you, John. Sorry.
Okay, thanks, Des. And so we fulfilled and placed all the tags across our application here. And now we're ready to do an analysis. So let's hit calcs. And all this is done on board within our application. Nothing sent outside, nothing has to leave. Not like some of the other technologies that are sending things to cloud servers, etc. And now it's doing that shape analysis, finding best matches across shapes, across our catalog of a meticulously analyzed MRI library. And it presents us a 3-dimensional volume of this case of RV end diastole.
So if I may, Jim, so what you are seeing with those points on the screen are all of the points that were dropped by the user as they work their way through the views.
Okay, and so we have our end diastolic tags: pulmonary, tricuspid, RV apex. We can see the tag we placed for the RV apex. We have some features to make it more appealing, easier to understand and see. We have a combined view, end systole and end diastole. And some other key features we have are, say, our scan plane overlay view. So here, I click on a particular anatomic tag, and it presents me with the view where the 2D view where this tag was placed, but it also puts it into perspective of how the volume, the overall volume of the RV relates to this particular 2D image plane. Just make it a little bit more apparent for you.
And you can also show multiple planes as well.
And then we can do multiple planes like Des is saying, so I can show a short axis plane in relation to the long axis plane. And I can put more on if I wanted. So it's a little hairy, but that's essentially what that's doing. So let me just put that back on for us, so we can get an understanding and a perspective of how the 3D volume relates to the 2D planes that the sonographers were acquiring. But most importantly are the metrics that are provided: end diastolic volume, corrected for body surface area, and systolic for body surface area, and again, the ejection fraction.
There's also a quality check that you can do, Jim, if you don't mind just swapping.
Sure. So we can go back to the 2D scan planes. Let me just bring 4 up. And this is the quality check that Alvira is describing. So each scan plane has tags on it. And what our software is displaying is something called borders. And it's telling us the conformational, or how well registered our analysis is compared to the 2D image that it was captured from to help generate the 3D volume. And we can see we've got very good registration all the way through. There's nothing way outside the boundary of a shape that we're not expecting. And we do that for end diastole, and we can do that for end systole also. So that's our sanity check, our quality check, you might say. And going back to 3D, and then inevitably, we're interested in the numbers. So we can also generate a report for that, a PDF report of the chambers that were analyzed. So we did do an analysis for the RV. We did do an analysis for the LV. Oh, I did it for all 4 chambers here. And then we can present that quality check imaging aspect for the report. So do we have any questions? Any high-level questions, low-level questions?
So thank you, Jim. Can you do this for the LV as well, and other chambers such as the LA and RA?
Absolutely.
For all the 4 chambers.
In this particular, we can do all 4 chambers. So again, here's our quality check. And when I hit the 3D, we can get a 3D volume of the LV. Aortic, mitral, of course, apex. Just make that. And we can do that for the left atrium. And the right atrium. So yes, all 4 chambers can be analyzed as the images are acquired.
It's quite a big step forward compared with what we've seen before.
yeah, we're very excited. It was not easy, but it wasn't hard. It was just hard work, is all it was.
And we really like to show
So
you what's coming. Show you what's coming.
I have a question. It's Bob Howard. I have a question.
Yes.
Go ahead.
There are some points, and on this image shows there are some points, like there's a point that's outside the final image. Yeah, that point there.
Yeah.
So, I mean, if that's a real point, I mean, if that point was put on a real border, what does that mean? We're smoothing then, right? Like, so it's not totally accurate, I guess, is my point.
Well, I think it's taking into account that maybe if you're not entirely sure of what that exact point is, the model actually, I wouldn't say tries to correct it, but would say, that point would be out of the realm of reality. So it doesn't necessarily follow and incorporate those points identically, because it actually goes back into our catalog and our library and our AI, and it says, you know, in theory, it is placed out of the actual volume itself, which is okay. Like, those points aren't denoted exactly on the border itself, because it's actually up to the AI to determine where it really should be.
And so, and again, our matching for shapes here, it's not a one-to-one match. So this isn't our three-dimensional volume that you see. It's not come from one heart inside our library. It's come from one heart with respect to specific tags in this specific area, and then another heart may have fulfilled the specific tags and found a shape, and then we marry all these regionally acquired tags together to present one three-dimensional volume. So it would take this particular pulmonary tag and this one and this one and this one and find the best shape that matches. And then it would also take this particular tricuspid analyst tag and this RV endocardial tag and this RV endocardial tag to help generate, find the best shape that matches, and then it brings everything together. So it's not a one-to-one heart by any stretch.
Understood. So how many images are in your library that you're using to actually do this?
It's in the order
and are you going to be?
It's in the order of hundreds to 1,000 max, somewhere around there.
So over time, are you continuing to add images so that your data gets even better? Because
actually,
instead of thousands or millions, or
no, because again, you know, just as far as AI and as far as Health Canada and FDA, the model cannot be changing because it always has to be verified and validated. So if you're always changing the core input data, you could be disrupting things in a negative way. So, you know, the rules are you actually have to freeze it. It cannot continuously learn, but we can, in addition, it would have to be like a separate release, which as far as the accuracy goes, we are happy with that at the moment. Not sure in the future are we going to be adding in another 10,000 images, for example.
But it also comes back to just the way that the way that the KBR works is it's taking
partial
segments, partials of different hearts, and the catalog has, as Desmond said, it has thousands of hundreds to thousands of hearts of different shapes and sizes that's supposed to cover basically a continuum of what you would see in typical clinical practice. And once again, it sorts through the catalog. If it doesn't find that particular segment, it moves on to the next one until it finds something together, and it puts it together. Almost the best way to describe it is like a 3D jigsaw puzzle to create that 3D model. So adding more hearts to the catalog isn't necessarily going to improve the algorithm.
Yeah, essentially, because we did capture diseased hearts, so pulmonary hypertensive hearts, primary pulmonary hypertensive hearts, congenital hearts. And those were, the source inputs were cardiac MRI images. So, much better resolution, temporal and spatial resolution images. And then those were analyzed with these tags. So when it's looking for tags from the echo input, it's comparing it against the tags from well-honed cardiac MRI tags, and then it's finding those partial shapes, and then we marry all those pieces together to generate this particular volume.
And
So
do you
one
have any
final question?
Yes.
Sorry, Chi-Ming, it's Bob. I had one
So, Bob,
final
go
question, which is, the number of tags that you can sort of accurately place must correlate with the sort of quality of the outcome of the final analysis. So there must be a minimum threshold that says, with this few number of tags, we can't really do this accurately. Do you understand my question?
Correct. And we do. There is a as we're proceeding through our tagging process. So what we gave you were the minimum tags, essentially in our template when we drag it across. And so this is sort of the minimum that we would like you to place as best as possible. And so 1, 2, 3, 4 at the apical level or papillary level. 1, 2, 3, 4, 5 at the distal LV.
Yeah, the idea is once you've placed all of the points on the different views that are displayed there, for example, for the RV, you have dropped the minimum number of points that are required, or rather the required landmarks that are needed for the algorithm to be able to generate the 3D model. But it comes back to, for example, on one view, if you're not able to see one of the landmarks, just move on to the next one that you've collected where you are able to see the landmark. And at the end, the software tells you when you've dropped the minimum number of points. And it'll allow you, that calcs button will be blue. Otherwise, it remains grayed until you hit that minimum number of points.
And doing something like this does not make it a lot more accurate, if you understand what I'm saying. Perhaps it might change it by 1 ml or half an ml or something like that when we're speaking about volumes. This does take a lot more work to put all these tags across, but it's not appreciably changing the resultant volume that comes up.
So if we have a study that's pretty bad, which happens, you would at some point you would say this, the algorithm doesn't work. We don't have enough tags here to do this properly. Is that right?
Perhaps. Just like you couldn't do a Simpsons on everyone unless maybe you were going to pour some contrast in to help delineate borders so you can track the border to do a end-diastolic and systolic 4-chamber, 2-chamber Simpsons. So sometimes we have to concede, but this is using, like Des is saying, sparse. And if we're seeing where we have a pretty good understanding of where tricuspid annulus is and pulmonary annulus is and RV apex, you place the tags and see what comes up. And so a lot of our formative validation trials were done with I would submit a difficult to image population of those adult and teen congenitals type of thing. Mm-hmm. And that's what our validation and FDA approval came out of, as compared to cardiac MR, that we were within that 9-10% variation, you might say. So it's it does its job.
So thank you. Can I ask a few questions? Number one is, can you do this with contrast? Like if someone give contrast to the picture, would that still work? You
Absolutely.
locating the points and borders?
Absolutely. Yep.
Yeah, yeah.
Because that just picks up your endocardial definition that much more. So when you're confidently placing those anatomic tags, it's going to be a much easier.
Maybe more confidence. Yeah.
More
So
confidence too.
Yeah. How much more time does it add to your workflow? Like, to do all this?
Acquisition workflow, minimal. Analysis workflow, 7 minutes more maybe, depending on how many chambers you're interested in volumizing, if you might say.
But it also comes down to as you get more comfortable with the software, we've seen people move through it very, very quickly.
Yes.
Yeah.
Wonderful. And last question for me is, in your AI model with all the MRIs, on the left side, on the left heart, have you taken into account all the really unusual disease stages, such as hypertrophic cardiomyopathy or dilated cardiomyopathy, all those kind of situations?
Dilated, sort of, yes. Hypertrophs, no. There's not a lot of hypertrophs in our database.
So the database.
Yeah, go ahead.
Sorry, Jim. Just to come back to how the database was developed, it wasn't developed for specific disease states. The idea behind it was to have different shapes and volumes from which the algorithm is able to pick them at different segments and put them together to create the 3D model. So it will sift through that catalog and find that particular segment that encompasses those landmarks that it's looking for. So it's not necessarily about the disease state itself, as it is more about the shapes and the volumes that it takes into consideration.